Hello, this is Dr. Hatami. Welcome to this video regarding functional anatomy of skeletal muscle tissue, chapter 10 of our human anatomy textbook. There are two types of muscle contraction. We have concentric contraction and eccentric contraction. Concentric contraction simply means when muscle get shorter, like when you pick up something or kick a ball. That is the example of the concentric contraction. Then there is eccentric contraction when muscle gets longer while contracting, like when you lower something down slowly or you're walking down the hill. Down the hill. Hello, this is Dr. Hatami. Welcome to this video regarding functional anatomy of skeletal muscle tissue, chapter 10 of our human anatomy textbook. There are two types of muscle contraction: concentric contraction and eccentric contraction. Concentric contraction simply means when muscle shortens while contracting, and eccentric contraction means when muscle gets longer while contracting. The best example for concentric contraction is when you're picking up a book or kicking a ball. But in concentric contraction, when you are going to lower something down, that is the example of the eccentric contraction. The mechanism for concentric contraction is less understood. In this video, I explain how concentric contraction works, which is called the sliding filament mechanism. Okay. To really understand this sliding filament mechanism, it's important that you know the structure of the sarcomere. Sarcomere is the functional unit of a muscle cell. This includes understanding the anatomy of the thick filament and thin filament. I already explained it in another video, and I'll share the link in the caption of this video so you can review it before diving into the sliding filament model. Hopefully you know the structure of the sarcomere and thick and thin filament. Now let's get started. For the sliding filament mechanism, to initiate this mechanism, we need the calcium release. Calcium is an important ion, which without calcium, there is no way that the muscle can be contracted. So calcium ions going to be released to the cytoplasm of the muscle cell from the organelle is called sarcoplasmic reticulum. We learned before sarcoplasmic reticulum is the modified smooth endoplasmic reticulum in the muscle cell, which they are responsible to store and release the calcium ion. Okay, so when calcium ion going to be released from the sarcoplasmic reticulum, now guess who is going to be the happiest molecule in the world? If you said troponin, you're absolutely right. Troponin would be the happiest molecule after calcium being released, because the troponin gonna be a bind to the calcium ion. Troponin is a protein found on the thin filament. After calcium binds to troponin, troponin shape changes and it's going to move the tropomyosin out of the way. Tropomyosin molecule, it was the protein found on actin filament blocking the binding site on actin filament. Now after releasing of the calcium, the shape change in troponin, tropomyosin molecule shifts and expose the binding site on the actin filament. And now guess who's gonna be the happiest molecule now? If you said myosin, you are absolutely correct. Myosin was waiting for this moment for a long time. And then now the myosin heads of the thick filament bind to the actin. And we call this as cross bridge. So cross bridge formation happens after calcium release, troponin changes shape, and also the shifting of the tropomyosin out of the way. And now when the actin filament, the actin molecule has been naked, and then the myosin heads bind to the actin. Now the myosin heads of when they are super happy and super excited, they can rotate inward and then pull the thin filament toward the center of sarcomere. We call this movement as power stroke. So power stroke is the movement of the myosin heads toward the center and pulling the thin filament toward the center of the sarcomere. Okay, now the ATP binds the myosin and it's time to be detached from the actin. So at this time we say the cross bridge has been break down. So then the myosin heads return to its original position and the cycle repeats and repeats and repeats and repeats. Pull the thin filament toward the center of the sarcomere. That is something going to happen during the sliding filament mechanism. Okay, now let's practice more by drawing the sliding filament mechanism. The first thing that we are going to draw is to draw the actin filament. So as you can see, we have some actin molecule here, and then we should have the myosin. Just imagine that right now we have calcium in the cytoplasm, and then we have the myosin head attached to the actin. So we can call this step as attached step or attached state. When myosin and actin bind together, we call this as the cross bridge. Okay, what's gonna happen next? So again, we should have the actin filament or actin molecule. And next, right now, after binding the myosin and actin, myosin is full excited, full of energy, and then it's gonna like bend and then move the actin toward the center of the sarcomere. After that, it's getting a little bit tired. So let's name this step, which is the power stroke state. When the myosin is gonna like move the actin filament toward the center. That is getting a little bit tired, and then right now the ATP gonna be attached to the myosin head and says, you know what, you need a little bit energy. Let's be detached from this actin molecule. So right now, then it's gonna be the detached state. Let me just make the myosin molecule a little bit depressed because actually it does not like to be kind of away from the actin. So it's, as you can see, it's a little bit depressed. So right now we know the ATP has been bind to the myosin, and then we have the detached state. Then we have breaking down of the ATP. So it means that the ATP is going to like release the energy, and then the myosin gets back to the original position. then it's gonna be bind to another actin molecule, and this is gonna repeat over and over and over and over. We have attached state when there is calcium in the cytoplasm, the myosin head and actin would be bound together, and then we have power stroke when the myosin is super excited, full of energy, and it's going to pull the thin filament or actin toward the center, and then it gets a little bit tired. Then the ATP bind to the myosin and get, hey, let's get a little bit like relaxed, and then it gets energy more, get back to the original position, and then it can bind to another actin molecule. That is the cross bridge cycle or sliding filament mechanism. So if you look at this picture, is very helpful to look here. You can see when the myosin attached to the actin filament, when the actin is naked right now as tropomyosin shifted, and then these binding sites are exposed to the myosin. Myosin and actin bind together, make the cross bridge formation. Look at this movement, which is called the power stroke, and look at the movement of the thick filament toward the center of the sarcomere. Contraction also changes the striation pattern in the skeletal muscle, which we can see when we look at the sarcomere in both its relaxed and the contracted states. In a relaxed sarcomere, the thin filaments only partially overlap with the thick filament. So we can see the I band, the area that we only have the thin filament, and then we have the A band, which is the length of the thick filament, and also we have the H zone, which is the area we have only thick filament. And look at here, the fully contracted sarcomere, when the muscle contracts, something changes. You can see the Z disc move closer together. What does that mean? It means that the sarcomere shortens. And then as the Z disc move inward, so the I band shortens. You can just compare with the fully relaxed sarcomere. The I band shortens, and also the H zone disappears. There is no H zone anymore. But look at the A band. If you just measure the A band or compare it with the fully relaxed sarcomere, you can see that there is no change of the A band length. A band stays the same because the length of the thick filaments does not change. The changes in the bands are due to the increased overlap of the filaments as the actin filaments slide past over the thick filament. So there is not because they are shortening themselves, because just we have the overlap between the filaments. So we know thin filaments or actin filaments slide over the thick filaments. We learned that filaments do not shorten. There is no changes of the length of the thin filament and thick filament. We saw that how the Z discs move closer together and shorten the sarcomere while muscle contracting. I bands shorten due to the increased overlap of thin and thick filament, and then H band disappears as the overlap increases. So now we do not have an area that only the thick filament would be available. Then A band remains unchanged because the length of the thick filament does not change. Thank you so much for watching this video. If you have any question or any thought regarding the sliding filament mechanism, feel free to comment down below. Thank you.